Hey everyone, welcome to Practical 2. So, before we actually get our hands dirty in the lab today, we need to unpack the absolutely fascinating world of economic crops. Think of this explainer as your quick roadmap. We're going to map out the fundamental plant systems, break down the crucial differences between monocots and dicots, and really explore the hidden architecture of angiosperms. We've got a lot of ground to cover, so let's just dive right in.
Okay, take a look at these two distinct young plants. On one side, we've got a maize seedling, and on the other, a cowpea seedling. Even at this incredibly early stage of life, they look completely different, right? But before we get into the nitty-gritty of those differences, we actually need to ask the most fundamental question for this entire practical. What exactly constitutes an angiosperm? It's a totally foundational term in the botany of economic crops. And really, getting this down is key to everything else we're going to uncover today.
So what is it? Well, an angiosperm is, quite simply, a flowering plant where the seeds develop entirely enclosed within a fruit. But the real crucial distinction, that comes down to the seeds themselves. Take the maize grain, for example. It contains a single cotyledon, which is really just a fancy word for a single seed leaf. Because it just has one, that makes it a monocot. Now, look at the cowpea seed. Notice how it neatly splits into two symmetrical halves? Each of those halves is a cotyledon, making it a dicot. And to put this into perspective for your agricultural studies, monocots and dicots together make up about 97% of all flowering plants on Earth. I mean, they are literally the absolute foundation of human agriculture. Now, as these plants start to germinate, those hidden seed differences suddenly become our absolute quickest field identifiers. The maize seedling, our monocot, pushes up this single, narrow first leaf. It's perfectly mirroring its single hidden cotyledon. On the flip side, the cowpea seedling, our dicot, unfolds two distinct, rounded seed leaves before its true leaves even start to show up. For an agriculturalist out in the field, spotting these emerging seed leaves is a total game changer. It is the absolute fastest way to categorize a plant, long before you ever have to dig up a root or wait months for a flower to bloom.
All right, so moving on to the bigger picture. Every flowering plant organizes itself into two totally distinct worlds, divided right at the soil line. Above ground is the shoot system. You can think of this basically as the plant's solar panel and reproduction network. It's got nodes, which are these vital traffic intersections where leaves or buds pop out, and inner nodes, which are really just the empty highway spaces between those nodes. And of course, you've got the buds themselves, just waiting to develop into brand new shoots. Then, below the soil line, we have the hidden half, the root system. This is made up of the primary root diving straight downward and secondary roots branching off to anchor and feed the whole shebang. Keep this dividing soil line firmly in mind, okay? It's the ultimate anchor for understanding how plants literally engineer their own survival.
Let's actually slow down for a second here and really look at root classification, because it's based entirely on how and where roots develop. I want you to pause and predict something for me. Based on the seedlings we saw just a minute ago, which of these three root systems do you think belongs to a grass plant like maize? Got a guess? All right, let's break them down. First up, the taproot. Like the cowpea, this features one dominant root driving straight down into the earth like a heavy spike. Second, we've got the fibrous root, and yes, if you guessed this is what you'd find on maize and other grasses, you hit the nail on the head. It's a dense, shallow network of thin roots that are all roughly the same size. And finally, the adventitious root. Notice how these roots, like the thick prop roots you see on maize or sugarcane, are emerging directly from a stem node above the soil surface. They aren't coming from below ground at all. That difference in origin is a super vital distinction to remember for crop management. Okay, zooming in way closer now. Let's examine the actual surface of these roots. See those fine little extensions? Those are root hairs. And what's absolutely fascinating here is that each one of these hairs is actually just a single, incredibly elongated cell. Literally one cell. Their primary job is to dramatically increase the root's surface area, acting like microscopic sponges to maximize the absorption of water and minerals. But notice exactly where they are. They're highly concentrated in a specific region called the absorption zone. They actually don't run the entire length of the root. I want you to start wondering about that right now. Why exactly wouldn't a plant want these super absorbing hairs covering the entire length of its root system? Keep that in the back of your mind.
Now, right down at the very tip, the root has to perform two vital, simultaneous jobs. First up is the root cap. Think about it. Microbes, rocks, tightly packed dirt, soil is a really abrasive environment. It's basically biological sandpaper. So the root cap acts as a tough, protective helmet. It has one single mission: shielding the delicate growing tip from friction and mechanical damage as it literally pushes its way downward through the dirt. And then, immediately behind that protective shield, we have the zone of cell division. This is where cells are multiplying incredibly rapidly to produce the new tissue required for the root's continuous downward drive. So the cap protects, and the division zone pushes. It's an amazing little system.
You know, every complex root network and sprawling shoot system we've discussed today originally traces its entire life back to just two tiny embryonic structures. First is the radicle, which is the embryonic root. This is the very first structure to emerge during germination, actively growing downward into the soil to establish the plant's lifeline. Second, we have the plumule, the embryonic shoot. This emerges shortly after and pushes upward toward the light. Every towering stalk of maize or massive cowpea vine begins right here, with just the radicle and the plumule.
All right, let's quickly recap the core points before you head out and get your hands dirty. Remember, monocots have one cotyledon, dicots have two. The shoot system commands the space above the soil line, while the root system colonizes below. Tap roots, fibrous roots, and adventitious roots are classified by their distinct structural origins. Root hairs act as single-cell sponges for absorption, and that tough root cap acts as a helmet for the growing tip. Now, before you step into the physical lab, I want to leave you with two structural mysteries to solve. Question 1, both stolons and rhizomes give rise to new plants, but why? And question 2, what is the true function of those root hairs, and why on earth don't they extend the entire length of the root? Think about how beautifully these plants engineer their own survival. Write down your hypotheses and bring them with you. I can't wait to see what you uncover in the lab.